All right. Good morning, everybody. We'll start on time. So my name is Dao Nguyen, and I'm the director of the McGill AMR Center. So it's a real pleasure to have you today join us for the third in-person annual AMR Research Day that we have. And so for those of you who are joining us for the first time, and you're wondering, who is the McGill AMR Center? I've put up for you the faces of all of us who are on during the year and working on making the place run. And in particular, I'll invite you to meet some of them today, as well as Angela Casey, our program manager extraordinaire, if you have any questions or wondering where you should go next. So today we're as a center, we're on to our third year, and so we've been working on her and growing up. So what does that mean exactly? So I thought that just as a starting to our annual symposium, I'll just give you a whirlwind tour of what has happened in the last year. So first of all, if you haven't noticed, we have a new logo, which is, a big upgrade from our ugly one previously from the McGill University. We've been working hard on also fundraising and bringing in resources for the members. So far we've brought up to $4 million over the next 5 years to fund some research, seed funding to help develop resources and support education and outreach activities from the center. We've worked in the last year with a variety of different groups within McGill to support research and create partnerships. In particular, we've partnered with the McGill Innovation Fund to create an AMR award to support teams that have intellectual property and technologies that can be translated into AMR solution. And so in the last year, it was exciting to be able to provide 2 awards. One on a novel therapeutics, a small molecule that can sensitize intracellular bacteria to host-mediated bacterial killing by macrophage, and another one to develop some real-time monitoring of airborne and waterborne microbes. So we've also partnered with the Centre de Recherche en Biologie Structurale for their Blue Sky seed funding, and we just recently awarded another team for their collaborative work on mycobacterial tuberculosis therapies. We've helped teams for co-funding of externally funded projects, in particular with the Genome Quebec. And then, it's the end of the year. I think we all work hard. And so I'd certainly like to put out the kudos to the teams that have been recently successful and things that I think are really quite innovative. One, Jérôme Waldischpohl that has successfully been recently awarded for us a big grant from Genome Quebec and Genome Canada. If some of you who were here last year, you might remember his video games and how to do citizen science and crowdsourcing to annotate metagenomes and antibiotic resistance genes. Another one from Makeda Semret and their CIHR, a first, as far as I know, of a antibiotic stewardship intervention in the ICU and using diagnostics and algorithm to try to limit the inappropriate uses of antibiotics in critical ill patients. And then for those of you also who were here last year and heard Yves Brun and Anne Marinier from the University of Montreal, they were recently successful with the Canada Biomedical Research Fund and BRIF to bring in some AI and high throughput microscopy enabled technologies to do drug discovery. Maybe a trend here is if you present it here, the next year you're going to be very successful at grants, perhaps. Otherwise, we've also been really excited to launch some training support. So we'll be welcoming our first clinical infectious disease fellow in antimicrobial stewardship come July. We've had a fantastic outreach team by students, 4 students in this year. And then last but not least, just last Friday, we closed in a EDAR 7, which was an international conference that was led by Dominique Frigon, one of our executive member. on the environmental dimensions of AMR. And it was a great conference here in Montreal that attracted over 400 attendees from over 40 different countries, and was co-sponsored by the Public Health Agency of Canada. And so that was sort of our year, and I hope that we'll continue to grow and have lots of new programs to bring to you in the coming year. And so in the spirit of collaborations and in breaking down silos, which is really what our center is about, this year's symposium deserves a few special mention. First of all is welcome to all of you who are not from McGill, that are coming from other universities. This is the first time we're opening the doors as widely. So I hope that you'll have the chance to meet new colleagues and for us to hear about your work. We have today, I think, over 200 people registered, and an exciting program of a keynote speaker with Kate Baker, which I'll introduce you in a minute. We have also a theme around genomics and evolution. Not all of the talks are about that, but we figure that after 2 years of the breadth and diversity of AMR, we were going to rein it in and try to dig a little bit deeper. For the program today, we also have 6 short talks by faculty members from McGill, from University of Montreal, Laval, and Sherbrooke to just give you a taste of their work. We've introduced some flash talks by students, so 14 of them over the morning and afternoon, and 45 posters for you to go and see this afternoon. And then for all the trainees at lunch, we have a special career insight event where we have 6 guests from outside of academia, from industry to government to nonprofit research funding organization. And then at the end, I hope that you'll stay for a reception at the end. So without further ado, let me introduce you our keynote speaker, Dr. Kate Baker, who comes to us from England. And so she was originally trained as a veterinarian and worked I didn't know there was critical care of small animals in Australia, and then moved to the UK to do her PhD at the University of Cambridge, and worked at the Zoological Society of London on viral zoonosis in fruit bats. And then she went on to do her postdoctoral fellowship at the Wellcome Trust Sanger Institute, where she worked on large-scale genomic epidemiology of enteric pathogens, particularly Shigella, and on antimicrobial resistance. And then in 2016, she's joined the University of Liverpool to start her own group, and where she held the chair of applied microbial genomics, and where she was also, I guess, having a double life in Cambridge, and was leading the prokaryotic research at the Department of Genetics in Cambridge. But then in 2023, has moved definitively now to the University of Cambridge. And so her research group, which I thought was very aptly called the Bakery, combines genomic epidemiology, laboratory experimentation, mathematical modeling to study enteric bacterial pathogens, particularly Shigella, and AMR in high and low and middle income countries. And beyond her research, she actually also has worked on an impressive number of projects and initiatives, from working with public health agencies to deal with Shigella outbreaks and evaluate and implement genomics into surveillance programs. She leads the Genomics of AMR Surveillance Working Group as part of this think tank called CEDRIC, Surveillance and Epidemiology of Drug-Resistant Infections Consortium. She advises the UK government and also serves on the WHO Technical Advisor Group on vaccines for AMR. So with that, my pleasure to welcome Kate.
Thanks so much for the introduction, Angela, and thanks to the organizers for the invitation to speak here today. It's a real privilege, for reasons which will become obvious over the course of the talk, to come to McGill in particular and to speak at the AMR Symposium. So as Angela outlined, I'm going to talk to you today about kind of the last decade of my research that's been focused on Shigella. So it spans both my postdoctoral work, fellowship, and the work of the group to the day. because it's been really transformative for us, having had this perspective of genomics on Shigella over the course of about, over 100 years now. And we've seen it go from intrinsic resistance to extensive drug resistance in that time. And I'll tell you a bit about the journey that we've had. So it's a talk in 4 parts. So shigellosis, which is what we call bacillary dysentery now. Dysentery is a syndromic term we use for when you have blood in your stool and severe diarrhea. And we now know that it's caused by both bacteria and amoeba, and essentially shigellosis is the bacterial version because it's caused by bacteria of the genus Shigella. And essentially, it was a pain right from the start. It was always a really bad pathogen. And then in the second part, I'm going to take you through how we've used historical isolates to really enable us to understand how it's the diversity and the adaptability of these pathogens that have helped it really stay a problem. I'm then going to touch on some of the antibiotic era things that we've done since we've started to be able to use genomics to understand this pathogen better. And then finally, talk about the methods we're using now to try and unlock the power of that to tell us more about the evolution of antimicrobial resistance in Shigella and other pathogens. So it was a pain right from the start. So where we are today with shigellosis is that it remains a really big public health priority. So it's the leading cause of diarrheal illness in children under the age of 5 in lower to middle income countries. And the case burden is huge. So you can see these numbers here. So 200 million cases a year about, and 200,000 deaths per annum. And unfortunately, most of those deaths do occur in that childhood demographic in lower middle income settings. So this is a little schematic that shows you the distribution of the disease. So you can see the target symbol underneath the map is scaled by incidence per 100,000 people. So there's really a lot of disease in Asia, and then still significant amount of disease in Africa, Latin America. And then that tiny blue dot in the middle is the incidence of disease across all of the high income nations in blue. So it's a really small proportion of the disease. And actually most of that disease is from people who've traveled to endemic regions and returned with the disease. It's becoming increasingly antimicrobial resistant, like many other pathogens. So this is just a graph showing you the y-axis is the number of antimicrobial resistance genes per isolate of Shigella. This is a particular subtype, but the trend is similar across all. And then you can see over time how they've really gained an excess number of resistance genes. And that red line just tracks the clinical implementation of the use of antibiotics in the early 1940s. And you can see that they've basically just adapted. So where we sit now with the Shigella kind of AMR perspective, some of you will recognize this as the WHO's recent release of the updated AMR priority pathogen list. And so this is really what the world is saying. These are the big deal bugs. And Shigella now sits here. So it had a bump up actually because it's become really more resistant in the last kind of 5 to 10 years. So it's both in this high group and now also in this critical group. And because of that really high burden of disease and the increasing antimicrobial resistance, the fact that we still don't have a licensed vaccine for reasons we'll go into, it's a priority organism for vaccines for AMR, which is shown in this figure here. So this is a report from the Wellcome, previously called the Wellcome Trust, so it's just Wellcome now, done with Boston Consulting Group. And Shigella is this, I have a pointer apparently. Yeah, Shigella is this guy here on the right, which scores big on disease burden and feasibility. And so there's a lot of momentum now to create a vaccine for Shigella. But this part of the reason that that's so challenging is because they're really diverse and adaptable. So just to give you, I'm going to try and keep the genomics light because I know it's a mixed audience. I'll tell you just what you need to know, which is that Shigella looks like this. So it's basically an E. coli. It emerges from E. coli in multiple places in the tree, so it's polyphyletic. And the bacterial chromosome is not so different, and it's about the same length and some of the same similar features. Because the real action for Shigella is in what we call the accessory genome, so everything that occurs outside of that chromosome. We've got lots of different virulence bits, which give it that severe disease syndrome compared to E. coli. A large virulence plasmid is what facilitates invasion of the intestinal epithelium, and then also a number of virulence islands on the chromosome. And then it's got a whole host of other mobile genetic elements that carry key functions, including antimicrobial resistance. So just to give you an overview of that, this is a bigger version of a bacterial cell with lots of wild and wonderful ways that you could have resistance encoded. So you can have mutations in drug targets on the chromosome, which are then kind of inherited Darwinianly and selected for. And then you can have integrative conjugative elements and plasmids of different types, with their own ecologies. So single copy large plasmids, or maybe multi-copy small plasmids that, dip in and out a little bit. So it's a very rich kind of flux for the accessory genome of Shigella. And the other feature that they have, which I'll not talk too much about today, but just to kind of draw that distinction with E. coli, is that they've got hundreds of insertion sequences, like literally hundreds, 400 to 600 per genome, distributed throughout their chromosome. And so these are just repeatable elements that move about somewhat stochastically, but then offer this opportunity for selection. So it's a really plastic landscape of genomes. It's not like kind of SARS-CoV-2 where it's not, there's a mutation. It's really, there's a lot of changes happening in genetic content. But of course, we didn't know any of this back in, pre-1900. So it was only in 1897, actually, that they managed to distinguish bacillary and amoebic dysentery. And so you can imagine this time in the past where, you had these explosive diseases, explosive outbreaks of disease from this really severe diarrheal pathogen. We had no idea what caused it. And it really was, as William Osler says, one of the great four epidemic diseases of the world. So William Osler was a very famous Canadian physician who was one of the founding fathers of Johns Hopkins Hospital. So the implications for dysentery at this time were really significant. It was actually thought responsible for the failed first attempt to build the Panama Canal because the outbreaks were just so severe they weren't able to keep a workforce in place. And that was until 1898 when Kiyoshi Shiga, a Japanese scientist at the Kitasato Institute, finally isolated this bacillus. So this is Shiga's bacillus, later known as Shigella dysenteriae. And only 2 years later, a German scientist, Simon Flexner, discovered Shigella flexneri, which was a different subtype. So they didn't serologically cross-react. And so this was in the time of the other great microbe hunters. And, this was when germ theory was emerging as a kind of commonly accepted phenomenon. And there was this great period of discovery where they were pulling out all these different types of Shigella. And then, of course, World War I happened. So this is all in this landscape of microbiological discovery and trying to finally understand the causes of all these clinically important epidemic illnesses. And dysentery actually outstripped typhoid and cholera as the most important pathogen on the war fronts. So it really caused these terrible outbreaks of disease, and actually continues to cause disease in war zones. The army are also really interested in Shigella vaccines because it's so contagious. It only takes 10 organisms to cause disease. And you can see this quote here from Robert Koch really underlining the importance of it in these settings. And war bacteriologist William Hoffman commented that during the war that there was these distinct outbreaks of both Shiga's bacillus and Flexner's bacillus, but actually various types of bacilli were being found. And so this is a really key pathogen that at the time they were trying to, this was the time that kind of diphtheria and tetanus vaccines were being invented. So everyone was trying to get to a preliminary vaccine so they could, disseminate it in the war. They didn't succeed in that despite attempts, but what they did note at the time was that the opportunity presented by the recent war for a comprehensive study of the dysentery group is unrivaled. And so it's become possible to collect a series of representative strains on a scale which has never been attained before and is unlikely to again. And so this was Andrews and Inman, who were two bacteriologists involved in trying to develop some of these early vaccines. Unfortunately for us, the Medical Research Committee, which became the Medical Research Council, our major funder in the UK, agreed with them and actually gave them a substantial grant to collect those diverse dysentery bacilli and to really create this centralized collection. And that collection actually exists to this day. So in 1920, they were given the money in 1919, and then they started this national collection of type cultures. Now, this remains part of the Public Health Service in the UK, and it's equivalent to the American type culture collection. So it's a dynamic and still growing collection of type strains, which are used both for, quality assurance across diagnostic laboratories, as well as for deposition of strains that are relevant to public health. So you can still call NCTC and have one of your strains deposited if you think it would be a valuable resource for the community. And so it's been running since 1920. And what did we learn from those early strains? We'll go into in a minute. But I just want to give you a hint of how far we've come since then. So these serological typing schemes is really the core of how we then went on to identify and subtype Shigella for years to come. So not only is it important for vaccination, it's also really important for tracking disease and stopping outbreaks. And so what you've got on the bottom left here is the start of a small timeline where we're looking at these different subtyping schemes that have been tried for Shigella. And you can see there 1915 and 1918, which is those early serotyping papers. And we actually haven't come so far. So, 15 years ago, this is still our major understanding of Shigella. So we've got these 4 species, Dysentery, Flexneri, Boydii, Sonni. Flexneri and Sonni cause about 80% of the disease, so I won't touch on the other ones today. But really, you've got then within these different serotypes, except for Sonni, so you've still got really diverse pathogens. So this is actually quite a hard target to get as far as tackling disease goes. In terms of other subtyping things we've tried, this is the rest of the timeline. So actually, we've tried various things to subtype Shigella. So we've tried phage typing, Collison typing. You can see from the 1980s onwards, after the kind of advent of the era of molecular biology, this massive explosion of techniques. But like my mom always says, if there's lots of different ways to do something, it's usually because one of them is not very good. And so this is kind of where we were when I started entering the kind of space of bacterial genomics. And actually what happened then, of course, is genomics has now come to the rescue. And so now using large collections, we've managed to create these subtypes of these species with lineages for Shigella sonni. We've got, I think they're called clades for Dysentery, clades for Boydii, and phylogroups for Flexneri, because heaven forbid we should have called them all the same thing. But the point is that we can now really exquisitely subtype these pathogens, which we've been tackling for 100 years without much of a way to really get at transmission chains and genetic content that's really important in addressing them as a disease burden. Now, I'm going to leave that thought with you, and we'll come back to that in a minute, because I want to keep you in the past just a little while longer and help you understand how we've used these historical strain collections to really help us understand how this diversity and adaptability have maintained Shigella's position as the top cause of disease. So NCTC1 showed that actually Shigella was off to a really good start. Now, NCTC1, unimaginatively, is the first isolate that was accessioned into the National Collection of Type Cultures, hence the name. And you can see a vial of it. This is one of the earliest vials of it over here on the left. Now, this was a Shigella flexneri type 2A. I won't get too hung up on the nomenclature of these because there's lots of different ones, and they're all acting relatively similar. And on the right-hand side there, what you can see is the accession card for NCTC1. Now, this bacterium was isolated in 1915 on a battlefield in Flanders. Now, that was 14 years before the discovery of penicillin. And so this had never seen penicillin, had never been selected for. But what you can see under that red arrow is that actually it was already resistant to penicillin and azithromycin when they pulled it out the freezer and phenotyped it after these molecules were invented and started to be used in the clinic. And this intrinsic resistance is actually consistent with our now knowledge that, actually these AMR genes evolved long before we even came on the scene. This was always bacterial warfare. Many of the antibiotics came from there. And so what it does, though, is tell us that the organisms were already off to a good start as far as AMR was concerned. So before I tell you a bit more about the science, though, I just want to flag that there's like an absolutely fascinating personal story attached to this, where within the frame of this study, we were able to pull out the details of the soldier who died from this isolate, because basically it was called the cable strain because patient confidentiality wasn't what it is now. And so if anyone's interested in following up on this guy who essentially otherwise would have been an unknown soldier, there's a really lovely video that we did associated with the papers on this. But what did it tell us? So scientifically, what did we learn when we were looking at these strains? So long read sequencing wasn't a thing 10 years ago. So it's very common now to be able to do nanopore and to be able to reconstruct full genomes. It's actually still quite challenging for Shigella because of these hundreds of insertion sequences. You get really poor reassembly of the genome. I'm good. I'm seeing lots of nods. This is good. So at the time when we did this study, this was a commemorative piece for the centenary of World War I. And there was only 4 strains that had been long read sequenced. And we did these on PacBio when it was a very new technology. And so what you're looking at here is a comparison between the NCTC1 strain, which is this 1915 isolate from First World War, And then the next closest strain we had in time, which is 1954. This is a very common model organism in the lab for Shigella. The red bars are showing you the comparison between the two. Red bars mean synteny, so you've got the same genes in the same order. And where the blue bars are there, you've got the same genes in a different order. Where you've got little gaps opening in these curtains and colored blocks overlying the 1954 genome, you've got the introduction of new genetic material. Now, this is all the new genetic material that was introduced in time. It's not selective. And when we drill down on the function of that genetic material, what we can see is that actually the introductions relate almost exclusively to the acquisition of virulence genes, antimicrobial resistance genes, and then there's this one other island of unknown function. When we fast forward to 1984, you can see that we see those virulence and antimicrobial resistance characteristics have come into the genome, presumably created a selective advantage because they've stayed in the genome. And we see that pattern repeated in 2002, where we get the introduction of these materials and it's stayed in. And the only other thing that was acquired in the 2002 was a serotype conversion island. So I won't go into too much detail on this, but basically Shigella serotypes can switch. So they're all the serotype encoding genes are on mobile genetic elements themselves. They're on phages and plasmids. And so what this does functionally is change the O antigen of the bacteria, which basically means it can escape naturally acquired immunity. And sure enough, this 2002 strain was described from an epidemic of a new serotype. And so these are really important functions for the bacteria. But what is that? So doing this study, you know, it was only 4 genomes, but actually this is evolving very streamlined. So it's just acquiring virulence, antimicrobial resistance. And it got me thinking, what is this other island that is as important as these other functions? And so the question became, can we use historical isolates to identify novel critical functions in Shigella? Now, the short answer to this question is yes, but the long answer is a really good story. So I'll just ask you all to kind of settle in, and we'll go from there. So in order to tell you at the end of this story, I'm going to have to introduce you to Everett George Doon Murray, who many of you might know. But he was a bacteriologist he wasn't a bacteriologist when he was born, but he was born in 1890 in Johannesburg, and then moved to do medical training in Christ's College in Cambridge, just down the street from my department, actually, and St Bartholomew's Hospital in London. He qualified in 1916 and then was very quickly recruited into a team to look at the outbreaks of cerebrospinal fever, which was caused by the pathogens of meningitis. And so developed a serological subtyping scheme for meningitis. He was then recruited to Mesopotamia for by the War Office and actually caught dysentery and convalesced in India. And on his return, he then developed this serotyping scheme. Those of you with really keen eyes might have seen that actually he developed the 1918 serotyping scheme for dysentery. And as if that legacy wasn't enough, he then also described the pathogen Listeria monocytogenes, which causes listeriosis. Again, building on really incredible legacy, he then built up the microbiology department of McGill University, and is actually probably why many of you are here today in a very indirect way. So it's really appropriate that his legacy should come up today. But there's another really important aspect of his legacy that's allowed us to learn about Shigella, and that is the Murray collection. And what this is that over the course of his life, he collected Enterobacteriaceae from 1917 through to 1954. And you can see here an excerpt from his famous dysentery notebook. And I hope those of you, again, with keen eyes might see the name, a familiar name under here of Cable Y, which is our Ernest Cable NCTC1 strain. Now, the important thing about this collection of Enterobacteriaceae is, of course, it covers the antibiotic era. And so we've captured pre-antibiotic era pathogens this is when penicillin was first used clinically. And just to, for those of you that don't work on Enterobacteriaceae, this is where Enterobacteriaceae sit on this WHO priority pathogen organisms list. And so it really is the historical backdrop for many of our most resistant strains that we have now and are facing today. And it provides an invaluable historical context. Now, unfortunately, E.G.D. Murray died in 1964, but a final really important part of his legacy is the fact that he had a son, Robert Murray, who also became an eminent microbiologist in his own right. Most of his career was spent in Western Ontario, but he also received an honorary degree from McGill. And so the strains were passed on from father to son on his death. And Robert Murray always recognized the value of these strains, and he tried to encourage people in his laboratory to get excited about investigating them, and was frustrated that he wasn't able to. And so he visited a conference in the UK and saw Peter Sneath in Leicester and said, look, we've got these strains, we need to be doing more with them, what should we do? And so Peter was very involved in systemic bacteriology, so taxonomy, and kind of started spreading the rumor around. And then it reached the ears of Professor Naomi Datta, who was at Hammersmith Hospital in London. Now, she's a titan of antimicrobial resistance, did some early work showing that it can be horizontally moved around. And there's a really lovely correspondence between everyone at this time, watching how they tried to make this happen. It was a really challenging thing to do. But as outlined here, suddenly there's a spate of letters, and it looks like this concerted effort to retrieve the cultures that my father had is going to be really good to do after years of not having made the best use of them. And Naomi writes back and says, after all this time, we're finally getting to do the work on your father's strains, and we're looking for the presence of plasmids in pre-antibiotic era organisms that don't have AMR. And so the question becomes, did plasmids come with AMR, or were they already there and acquired AMR? And those are exactly the studies that were done with the strains. And sure enough, they had 2 papers in Nature later that year that basically showed that it was the plasmids came first and the AMR got imported onto them. So this is an really important finding for resistance evolution. And the Murray collection's been used a lot since to look at Salmonella plasmids, other antimicrobial resistance profiles, mercury resistance. We did some work on Klebsiella virulence as well. Now's the part where I'm going to go a little bit meta on everyone and write myself into this story, like some sort of really bad Andy Kaufman adaptation, for those of you that have seen it. This is me as a young postdoc at the Sanger Institute. Actually, this is in the NCTC1 video. So I'd been working on NCTC1 and then heard about this Murray collection. Because actually at the time, my supervisors, Julian Parkhill and Nick Thompson, were accessioning it into the NCTC. So it had been brought over, Naomi had done the work, but the strains were yet to become widely available publicly. And so as part of that initiative, we sequenced a bunch of them so that they would be available, the genomes would also be available for study because of the importance of it for studying antimicrobial resistance. So you can see here the table on the left and the timeline on the right, just showing you the distribution of these strains. And again, you can see these really familiar and really important genus names on the left-hand side. So there's nearly 700 strains now accessioned into the NCTC, and about 400 of them are sequenced. There were some issues with having to do the high security strains at the time, but this is now being topped up as well. So please, if this sounds exciting and there's some organisms that you look, please do check out the original publication where all the strains are available and all the genomes are there. And actually, there's some preliminary analyses in there as well to help you guide strain selection. So if you've got a favorite model organism and you want to choose a closely related Murray isolate, then you can get that out of there and also look at the different types of resistance genes that they carry. And the point is that the strains can be got from NCTC as well, so you can actually work with them in the lab as well. So coming back to our question, so that was the long version. But can we use historical isolates to identify novel critical functions in Shigella? Absolutely. So within our lab, we do quite a lot of bacterial genome-wide association studies. So this is essentially where you take a collection of bacterial genomes and you'll have a phenotype of interest. So, you might choose severity of disease, so invasive versus non-invasive disease, or potentially you could be looking for genetic determinants that are associated with a continuous phenotype like biofilm formation or something. But essentially you can feed the genomes into the algorithm and say, what is associated with X? And we tried to apply this to time, so it's why it's called temporal GWAS. So instead of saying, tell us about how much biofilm they can make or whether or not it's associated with clinical disease, we said, what's associated with time? And we were only able to do this because of the Murray collection. So you feed in the first 50 years of data from the Murray collection and the next 50 years from modern strain collections in those early papers. And what comes out of that is a whole bunch of really exciting genes. So this isn't that intuitive a plot if you're not used to looking at them, but this is a bubble plot from the bacterial GWAS. Oh, now it's click, hang on. So what you're seeing here is a series of genes, which is the labels. On the x-axis is the effect size. So that's like 3 times more likely, 4 times more likely. And the y-axis is significance, statistical significance. So you're looking top right of the graph is the exciting bit, and the size of the bubble is just kind of the amount of genetic content. This is a KEMA-based analysis, so it's basically the amount of the gene that's associated. And when we looked at the function of these different genes that we'd recovered in this temporal GWAS, we found that we recovered virulence, which was really encouraging. We also covered antimicrobial resistance, super intuitive. But then we did also get some really exciting other candidates. So this massive plot, top right of the graph, unknown function gene. So we have no idea what that gene is doing, but it's even more associated with more modern strains than resistance and virulence. So a PhD student at the time, Rebecca Bennett, has pulled out this protein, done some 3D modeling of it. And the truth is we're still trying to figure out exactly what it does. What I can tell you is it was labeled as an adhesin, but we've now done further work. We don't think it's an adhesin. It's called Steve. This is as far as we got. But what we can tell you is that Steve is actually probably important in a lot of other pathogens as well. So this is a tree showing you the protein, the relationships of closely related protein families. So this is the amino acid phylogeny of the protein. And you can see on the left-hand side the different variety of organisms in which we found this protein. And the pink labels on the outside show you where the Enterobacteriaceae are. So it's really well distributed throughout this really high priority AMR organism group. So we did the discovery for Steve in Shigella flexneri. And then we wanted to look at it, we kind of to validate these results and to say, you know, because we were coming forward and saying, we think this is really important to the epidemiology of Shigella because it's associated in the same way that virulence and AMR are. Okay, you're going to have to use your imagination a bit, I can see, with these tree figures. But essentially, can you guys see any of that? No, okay. Pointer it is, right. Okay, so basically there's a very big tree here and there's a bunch of different lineages. And this one here, from here down, there's a kind of purple shading. This is lineage 3. So this is a tree of Shigella sonni. So this is our cross-validation. We've created, yeah, if we could have the lights down, I think that would help. There's lots more trees. So if there is a chance to get the lights down, that would be good. Anyway, so this is lineage 3, and this lineage 3, without going into too much detail, is taking off around the world. It's our big, emergent, nasty AMR strain of Shigella sonni. It's the one we're all worried about. And the reason why historically we've thought that it's so prevalent and distributing around the world is because of its antimicrobial resistance, which is shown by all these black blocks on the right-hand side. So the idea is it's got lots of AMR, it's spreading around the world, that must be why. When we actually go in and add in our Steve protein, which is this aquamarine little label here, you can see that Steve was actually acquired in this lineage well before antibiotic resistance. And so while we don't know what it does, and we're still getting to the bottom of this in the lab, we do think that it's going to be really important. And so the work continues on this. So as I said, the long answer is yes, we can use historic isolate collections to identify novel, potentially critical functions. And if anyone else wants to read more about what you can do with historical isolates, then do go and read the review paper. Okay, so we're going to leave the past behind us now for a little while, and we're going to talk about how genomics has really transformed our ability to track and treat Shigella. Because all the richness of like the trees and the lineage taking off around the world, that's actually the bread and butter of what my group does. So we do a lot of genomic epidemiology. We collaborate a lot with clinical and public health agencies. We do large collections and we correlate it with metadata. Because without genomics, that rich Shigella sonni tree that I was showing you, this is a single serotype pathogen. And so that's what that study looks like if you don't have genomics. It's just a big bin of Shigella sonni, and you can't draw out any of this. But when we take all the richness of the genomes and the mobile genetic elements and accessories, and we combine it with epi data, then we've really transformed our view of this disease. So for starters, we know now that lineages are geographically associated. So this is a study of 9 different countries in Latin America, where we worked with PulseNet Latin America, the public health surveillance network, and we were able to show through this is a radial tree, so it goes from outside to in, and basically where you see these black branches of the tree, that's the novel isolates from this study. The rest is historical backdrop. And you can see that we identified lots of novel sublineages associated with Latin America, and that ring around the outside just shows you the clustering by country. So really strong clustering by country, and you can really identify neighboring country outbreaks within others. And this scales to the continental scale as well. So this is a similar collection of Shigella sonni from different LMICs around Africa and South Asia. And you can see that isolates from Bangladesh, India, and Pakistan cluster together, and they're distinct from isolates from Africa, so Kenya, Mali, and the Gambia. And the important thing about understanding this geographical association is, first of all, you can track outbreaks across borders, but you can also use it to develop tailored treatment recommendations. So what you're seeing here is the genotypic prediction of resistance, which is quite valid in these organisms, clustered by region on the bottom and shown by antimicrobial class. And the fact that they don't overlay shows you that we actually need specific treatment recommendations for specific regions. And we can also use this to inform other treatment interventions other interventions that we might want to do for Shigella. I spoke a lot about the vaccine momentum, and actually because of this rich collection of data from LMICs, we looked at the variation in vaccine antigens. So what you're looking at here is a 3D model of one of the vaccine candidates, IPA-D, where we mapped on the antigenic variation and its impact on protein structure and saw whether or not any destabilizing mutations were found in epitope regions, which are the key regions for targeting the immunology. And so, you can really use these genomic technologies and data to do rich things for controlling it. One of the other things we've managed to do is identify new transmission pathways. So in the UK in 2009, there was an increase in sustained outbreak of Shigella flexneri 3A, which is a really obscure type of Shigella for England. We don't see it a lot. And it was really weird that it was sustained because it was in people who hadn't traveled, largely in males. So what you're looking at is the case numbers for males who hadn't traveled in the UK. And we had this massive uptick and it wasn't going away. So we started the study in kind of this time and they were like, why isn't it going away? These normally just come and go. And what we found, which we suspected, there had been a couple of reports of sexually transmissible Shigella. So this is a really highly contagious pathogen that's fundamentally fecal-orally transmitted. And so actually what you end up is you get minor amounts of transmission in sexual transmission networks, but this was sustained. So what we did was we took Shigella flexneri 3A from Asia, from Africa, and from patients who had traveled to Asia and Africa, and then a whole bunch of isolates from this UK outbreak, and then from outbreaks elsewhere as well. Because there was also male-associated outbreaks happening in Australia and Canada at the time, and France. And so what we did, once we sequenced all these isolates, we found that they actually all fell in this very low diversity sublineage of the tree, where you can see that there's very short horizontal distance for long vertical distance, which means it's clonally expanding very rapidly. And when we looked at the demographic features of the patients from this lineage, we found that over 95% of the isolates in the blue lineage came from men. And then there was a different age distribution as well. So you can see the proportion of isolates for each lineage by age group along the bottom there. And instead of having this cluster of very young and elderly people, we get a real peak around the middle age. And so this confirmed for us, along with interview data on behaviors, that actually what we had was the first really global outbreak of sexually transmissible shigellosis in lots of different countries. And so we can bring all these things together and look at the evolution of Shigella as a whole. Has that come out? Yep. Okay. So now, partly on the back of the early studies that we did in collaboration, UKHSA has turned on genomic surveillance for their foodborne pathogens now. So now every time someone gets Shigella in the UK, it gets sent to the National Reference Lab and it gets sequenced as part of routine outbreak investigation. So you're looking at everything that's happened basically since they switched on the sequences here. For Shigella sonni, this is. And you can see those same old signals. So I know it's a complicated tree, but I'll walk you through it. So this is the radial tree. There's about 3,500 isolates in here now. So things are really starting to scale. That first one is year, and you'll see that it's mostly a clonal replacement pathogen. So what I mean by that is year is on the heat map, and as you go clockwise, it tends to get darker. And so mostly we've got strains replacing strains. Then you've got age, sex, and travel, which are those key demographic features that we can use to pull out what these transmission pathways are. Ignore lineage and BAPs, they're just taxonomy. And then on the outer rings, those things in green are different antimicrobial resistance determinants. So what you're looking at with this orange wedge down here is a bunch of patients who had traveled to Africa, and you can see a mix of age and gender, and you can see when it was occurring. And so that's indicative of travel. So these people are coming back, and then there's a lot of kind of inter-postcode transmission where people are passing on to household contacts. We see a similar pattern over here with Asia, where we've got a strain where there's lots of returning travelers from Asia. And this one's associated with a key antimicrobial resistance marker. So QRDR is a series of mutations that give resistance against the first-line treatment for Shigella. That's what this little panel is here. These are all our things that are in the treatment bag. These are WHO recommendations for Shigella. And so now that lineage is completely resistant against QRDR, against ciprofloxacin. What you're seeing up here in this black arc is sexual transmission. So you can see when it moves from that travel in the blue and then moves into this black arc, you get suddenly very homogeneous age and gender. And there's very little travel and only travel to Europe, okay? And so you're getting this MSM-associated transmission, which then acquires resistance against azithromycin. Now, this was bystander resistance because of gonorrhea treatments, and then gonorrhea treatment switched from azithromycin to ceftriaxone. And then, sure enough, Shigella did what it does, and it just acquires exactly what it needs to survive the selective environment, and it acquires ceftriaxone resistance shortly after the pandemic. And this is what we're faced with now. So we now have extensively drug-resistant strains of Shigella. This plasmid the plasmid responsible for this has now moved into other lineages of Shigella. We know it's spreading worldwide. This study included Belgium and the US and Australia, and they're all in that final lineage. So one thing, if you take home one thing today, is that AMR is not a slow pandemic. We often talk about it as a slow pandemic, but for Shigella, we've gone from like susceptible to fully resistant in like less than 20 years. It's been about 15 years in the UK. So it's doing what it does, and its adaptability is really keeping its top spot. So I'm going to whiz through a couple of things we're doing to do. How long? Okay, 10. Okay. So things that we're doing to try and bring these two things together. Okay, so we've got this real richness of accessory genome evolution, and we've got these brand new genomic epidemiological frameworks where we really understand how the pathogen is moving and transmitting and the selection pressures it's under. So can we bring those two things together to tell some generic lessons about the evolution of AMR? So the things I'm about to focus on really rely on this model of convergent MSM-associated sublineages. So we already talked through this big black arc of Shigella sonni. Those of you with really keen eyes will be sitting there going, why hasn't she pointed out that there's another one here? And there's another one here as well, because what we do get in this setting is multiple lineages falling into the sexual transmission network, for lack of a better word. So we've got these multiple co-circulating sublineages. We've got this really high antimicrobial use because there's a lot of comorbid STI in this particular subgroup that are at risk of shigellosis. And these are actually, so it's quite high risk MSM subpopulations, so really dense transmission networks. And we've just recently done some work to show there's really rapid spatial homogeneity. So actually, for all intents and purposes, it's the same environment that these strains are trying to survive in. And so what that means on a public health level, or I guess from a biological evolution perspective, is that we've got diverse genetic backgrounds that are all falling into the same selective environment of really high antimicrobial pressure. So what we've learned from this, the kind of vignettes that I'll talk through real quick, that all AMR is not created equal, and that it's not all about AMR. And so as far as all AMR is not created equal goes, we see parallel AMR acquisition. So as I said, we've got co-circulating sublineages. What you're seeing on the left is 3 different types of Shigella and the co-circulating sublineages. So where the bits of the genome phylogeny are in color, these are different sublineages that we know are circulating among MSM. Now, they all need azithromycin resistance for the reason of that high selective pressure. So I won't go into detail about that. But what we found is that this azithromycin resistance that they all have is encoded on different plasmids. So 7 of the 9 had this purple plasmid, and then 2 of them had different green plasmids that encoded the azithromycin resistance in a different way. But you can see just from that the plasmid performance is different. So 7 of them had this purple one, only 2 had the green. And actually, when we did a review of the literature shortly after this, the purple plasmid had disseminated to lots of different countries, which is shown by the flags, cheers Canada, and lots of different subtypes as well. Oh, thank you. And the green one had never been seen again. And even at a finer scale, we see this difference. So just looking at this subtype S-Flexneri 2A, which is the bottom left guy, you can see that there's a really large outbreak associated with that purple plasmid, 16 cases a year, the bottom one, only 2. If you haven't followed any of that, just take my word that the purple plasmid is like better than the green plasmid. Okay, so what we did to try and discern what the differences between these were was we started the genomics because that's kind of our bag. And when you looked at the differences between these two plasmids, we could see the genes related to conjugation, AMR, and SOS inhibition. And so when we look at these in turn, we could see the difference in these phenotypes. So in different donor recipient types, so different types of Shigella and different E. coli, so we did this across different donors recipients, across media, across liquid and solid media, we found that actually the green plasmids were more conjugative, so which was counterintuitive, the idea that, we had this really successful plasmid which was less conjugative. But actually that's really helpful because it does tell us that actually probably the selection is more important than the stochasticity of the conjugation, which has actually been found in other model systems as well. We did find an increase in SOS inhibition in the presence of subinhibitory concentrations of antibiotics, which might be contributing to this as well. But the most marked difference we found was in bog standard fitness. So this is when you make a transconjugate and you see how well it grows. So the wild type is in gray, and then those transconjugates are in green and purple. And you can see that the green plasmid really has a massive fitness effect for these organisms. And so we think that actually this might be quite an important phenotype. The other important phenotype is that it has increased AMR. So it did actually cause more azithromycin resistance. But we've actually found, even with this ceftriaxone resistance plasmid that I described just before, this XDR strains, that also had a really low fitness effect. So within the group, we've convinced ourselves this is worth doing, and we've now got an MRC grant to really scale this up to a much more global level and to do this en masse across the mobile genetic elements. So watch this space for that. And then second lesson is that it's not all about AMR. So I'm just going to whip through this. So using the same system, we've got our MSM-associated strains, which we believe are under high selection for AMU as a proxy. And we can do bacterial GWAS to say what's associated with these strains that are being selected for. So this was an early study that was done as part of the original work. This is a Manhattan plot for those. I'll just walk you through it for those that aren't familiar with looking at it. You've got a reference genome up the top. Blues are the coding frames forward and reverse. The little gray dots are these genetic elements that are associated with this proxy for high antimicrobial use. And then the significance is on the y-axis. So where you've got these big kind of skyscrapers, it's where you have an association. Sure enough, this was our purple plasmid, which we kind of knew about. We've got another AMR determinant here. And then we have this signal of other, again, coming through. We've got other things that we're not looking for. And so this sounds familiar from our NCTC1 study. So I'm going to finish by saying, can we use this to identify stepping stones for AMR? And I will keep this to a short answer. So short answer is yes. So what we've done with this piece of work is we've scaled up that association for MSM-associated lineages. So now working with all the surveillance data from 2004 to 2020, we've identified our MSM-associated strains now using slightly more sophisticated methods. So we're using ancestral state reconstruction. We're then taking a subsection of strains, which is all the dots in red, and running what we're calling bulk phenotyping of epidemiological replicates. So rather than doing technical or biological replicates, we're actually saying if we want to know that lineage 4 is more whatever than lineage 5, we should be taking multiple lineage 4s rather than just doing one lineage 4 3 times over. So we're getting some really nice signals in the effects of some of our genetic determinants. Without going into it too much, but this is just growth by the copy number of these genes that we've found. And now we're actually just validating this by doing the mutant construction and testing. So I just wanted to highlight that this kind of general approach can identify novel genetic determinants that are associated with different characteristics of the bug in the presence of antimicrobials. So I think we need to harness these frameworks more to really understand the evolution of AMR. So just to summarize, Shigellosis has and it remains a global public health concern. And it's really its evolution that is allowing it to do that. We've used genomics to both create these epidemiological frameworks for directly tackling the disease as a public health burden, but also for being able to facilitate the discovery of new biology. And I really think, the historical collections have been instrumental in shaping these studies and also understanding how, AMR is evolving. So I'd really encourage people to get stuck in there. Massive thanks to everyone, particularly my group who did most of the work on these slides. And I think, especially today, it's been a really special experience coming and talking about the Murray collection at McGill. So I just really wanted to also acknowledge the work of these kind of titans of microbiology who really laid the path for the rest of us coming into this. And I think we've also privileged to have Bob Murray's daughter and grandson with us here today. So thank you very much for coming. And yeah, let's give them a clap. Thank you.
Thank you very much for this expansive presentation. We have time for questions. Anybody, there are microphones going around. If just raise. Okay. Maybe I'll start with a question. So you showed that with your green and your purple plasmids, you were comparing their fitness, and this is obviously lab-based fitness. Do you want to speculate a little bit? What are some of the underlying mechanisms and how that might translate into better fitness in the context of infection?
So we're not quite sure yet. So part of the reason, this is partly why we're scaling it up, right? Because it's n=2 at this point. And so what we need to do, I think, is first confirm that it's happening kind of at a public health level. But we do have one of the postdocs in the lab is really interested in getting to the bottom of this, like fitness, what is causing fitness, and actually taking a TRADIS approach to the plasmids. So knocking out all the genes systematically across the plasmids to find out what is driving that phenotype.
Hi. You hear me?
Nope.
Fine,
Nope. I mean, I can hear you.
Hello again.
Yeah.
Good. I just wanted to ask a question about your acknowledgement of Earth. If I understood, he wrote letters, emptied a freezer, shipped things to somebody, and was not a co-author of the papers. And is in some ways that's inspirational. In some ways, it begs the question of how do we do any sort of global research when we're asking people in some country to collect the bugs and other people to do the sequence analysis. So do you have any thoughts about the lessons from Bob Murray's generosity?
I mean, I think I'd like to think the way we're doing authorship is better. It would be extremely rare to have a Nature paper with 2 authors these days. And to be honest, having read through, I mean, I was given a box of correspondence this big associated with the strains to like draw out what had happened. And it's not just Bob Murray. There were, Rita Legros spent 3 months of her life. She flew to Canada and subcultured all the strains and came back and put them in NCTC, and she's not on it either. So I do, I think you're right. Absolutely. Like, should have been an author on that paper. But I think the way things were operating in those days is very different to how they're operating now. And so I think maybe it's actually a positive thing is that things have moved on. I don't know what we do about the kind of historical legacy of that practice.
Hi, do you hear me? Yeah. Thanks a lot. That was a very nice presentation. Lots of questions. In particular, something that stood out to me is that you showed very nicely that selection is a very driving force for these species or the genera in particular. But at the same time, you also mentioned that there are only 10 microbes necessary to transfer the disease. So it seems like genetic drift, the bottleneck through these transmissions, is also a very driving force. How much do you see drift, for example, the IS elements jumping into genes which are actually very essential for the disease, or in general diversity within a host?
So we don't actually look at the IS stuff a lot because overwhelmingly we work with Illumina data from routine surveillance. I guess the flip side of that is the other aspect of it is how we look at the isolates. So I'm thinking about public health visibility. So if IS elements do get into isolates and make them less severe, we generally don't see them because of the nature of our work. We're mostly collaborating with routine surveillance. So it's isolates that have made someone sick enough to present to a doctor and be referred. It's pretty good for Shigella because it's quite a severe disease, but I doubt that we'd see, kind of non-virulent forms. We do have in LMIC settings an ongoing piece of work around where we have actually captured that. We have isolates from controls who weren't sick at the time of presentation. So these were matched controls in a community case control study. And we're doing bacterial GWAS on that. And we've not really thought about the ISL, maybe that is probably our best bet, would be looking in that collection. So thank you for the idea.
Hi, thank you for the talk. I noticed you showed a lot of kind of outbreak scenarios where, or samples that were spanning multiple decades, or 2 decades, or 3 decades. Can you hear me?
Can you speak up?
Oh, okay. So I noticed that you were showing a lot of kind of populations that were spanning multiple decades in your trees. And I was wondering if there was any sampling effect in the genomic analysis, whereby let's say you were sampling at multiple points at constant time points, or it was just a random collection that was spanning multiple decades, and you would see like a certain difference in the genomic analysis, or in the presence absence of certain genes, or so on.
Yeah, so I think we really like working with public health surveillance data, partly for exactly that reason. So the kind of freezer study, is that what you're asking about? Is the impact of sampling bias on these studies? So I think generally, I mean, of course, with the pre-antibiotic era, there's nothing we can do about that at this stage. And it will have been very ad hoc. But I think the important thing is what they did was shape ideas that we can then take forward. Because I completely agree with you. You know, if we've got a bias or imperfect sampling, then it makes it very hard to draw inferences. There are a lot of modeling approaches now that can, you know, deal with that uncertainty, but it's not ideal.
Any other questions?
Thank you so much. Thank you.